These are the in-class questions for option B, musculoskeletal anatomy. And we've got a sarcomere shown here. And you should recognise that. And then we've got some sections through that. And whether A to C represent transverse or longitudinal sections. Well, a longitudinal section is a long. So longitudinal, it's a longit, whereas a transverse, trans means across. So if you imagine a banana, if you make a banana split, that's a longitudinal section. Or if you make circular slices of banana, that's a transverse section. So these are transverse. So they've gone chopped this sort of sideways. With reference to the micrograph, so we need to talk about this micrograph. For each of the circles, state the region of the sarcomere. So we're probably looking for names of bands and zones and things like that, and then some sort of explanation. Because it says, state which region, and then explain. So we need to do two things for each mark. And so A just contains small dots, B contains the big dots, and C contains a mixture of small dots and big dots. The dots are the proteins, so this is our actin thin filaments, this is myosin, our thick filaments, and this is actin and myosin. So that's our explanation forming. We just need to say which band. So for A, we are looking for something like this. So just the actin, just the thin filaments. So we need to remember the name for that, the isotrophic band or I band. And we just need to say it's the actin only. So only actin. B, that's our myosin, and this here is our M line. So these are Z's. I'll get rid of that A there because I confuse that with the A down there. That's the answer to our question. And this is our M line there. Okay. So we're looking at just the myosin. So this area here is just the myosin. So if we went, for example, there, that's just our myosin. So that could be that one. This could be this one. That's better. So just myosin, which is our, for example, our H zone. So thick filaments, myosin. So myosin, thick filaments only. And before we could have mentioned actin thin filaments. And for C, both of them, so we're going for this kind of thick part there. So it's the kind of overlap of the A band. So the A is all of the myosin, including where it might overlap. So both filaments is the key there. So the dark region of the A band, or the edge. So edge, dark region of A band. Why? Because it's overlap of actin and myosin. Overlap of actin and myosin. Thick and thin. Part 3, describe how the protein fibres interact to bring about the contraction of the sarcomere. So this is just to dump your knowledge on the page without writing too much, in just clear steps. And so the marking scheme does break it down into steps, and I'll put those A to F, but you just need 4 of these to get the 4 marks. And so let's start off kind of outside the story really, but let's talk about calcium and how that interacts with the first protein. So calcium ions bind to our first of the 4 proteins, troponin, and that changes its shape, changing its shape, and that has an effect on the second protein, tropomyosin. So that moves tropomyosin. So that moves tropomyosin, and that exposes the binding sites on actin. Exposes the binding sites on actin. So we mentioned 3 of our 4 proteins now. And then the business end is now we can have myosin forming cross bridges. Those myosin heads can now bind onto that actin. So myosin heads make cross bridges to the actin by binding to these sites that we've just mentioned. So it's just reeling off the theory from the booklet, and these are the sort of slightly more complicated bits. So saying about how this is powered. So our ADP and inorganic phosphate are released, and then that causes the myosin head to bend. So that's the thing where the muscle is actually contracting. And then we're just picking up the pieces now. So ATP joins to the myosin head, and that breaks the cross bridge. So sort of counterintuitive really. ATP joins to the myosin head, breaking the cross bridge. And then kind of an overall statement saying how the contraction happens with all of this put together. So the actin filaments slide across the myosin by this ratchet mechanism. So actin filaments slide along the myosin by the ratchet mechanism. So moving, releasing, moving, releasing, moving, releasing. So I know I've gone over the space there, but you only need to say 4 of those to get the marks. Part B, then we've got a tale about Tim Peake. And this is just really saying muscles get worse in space. And we've got a before and after shot, and clearly there's a difference. And then confusingly, this diagram here, they should have really put these a bit separate, but we've got our normal situation and our atrophic. So atrophy is kind of like wasting away. So our 2, so these are not connected, these are kind of the before and after. And if you look at these, we can identify our thin filaments, actin, and our thick filaments. So we've got, this is thin actin, and then we've got our kind of thick myosin. And we can see there that we've lost some of the actin. So we can see here we've lost some of the actin. There's normally a couple of strands, but you can see there's problems there. So we can see that we're not going to be able to form as many cross bridges that we just talked about if there's less actin. So that's going to be our answer somewhere. So part one, why is it important that the muscle sample before and after the flight is taken from the same muscle in the same astronaut? These are like controlled variables, or to keep it a valid experiment. Because some astronauts might be more muscly than others, for example. So the myofibrils could vary between individuals, for example. So myofibrils may vary between astronauts. And between, not much space there is there, between muscles in one astronaut. So we need the same muscle in the same astronaut. Using the image, so we've got to refer to this somehow, and your knowledge of muscle contraction. So using the image, there'll be a mark saying what the evidence is, and our knowledge of muscle contraction. So, affect the maximum force. So we need to answer the question. So the maximum force would be less. So maximum force would be less. And then why, we can see, we've got less actin. So the simplest answer is we've got less actin. Less actin. Or we can say, more spaces. And that's actually going to get us our 2 marks, but we can mention, so we're going to get fewer cross bridges forming. So fewer cross bridges forming. So reasonably straightforward, really. Part C, the bones of the skeleton can fracture. So we can see we've got a fracture there, and we've got a fracture, looks like a fracture there. Key bits of information. We've got C is our healthy 19-year-old male, and our other one is a 75-year-old female. Key bit of information there, suffering from osteoporosis. So we know about younger versus older people. We know about osteoporosis. So explain why the fracture shown in X-ray C is more likely to heal. So we've got an age difference and we've got a disease that we're going to talk about. So patient C, so patient C is younger and possibly more active. Younger people tend to move around more than old people. Not in my house, but generally. And D, we can say, I mentioned this osteoporosis and say our bit of knowledge about that. So D has osteoporosis, and that's where our bone is being built slower than it's being dissolved. So bone growth is difficult. So less bone growth osteoblasts are less active. They're the bone building cells. Osteoblasts are less active. If you've seen our excellent Holy Cross video on that. Part 2, and surgery, and they've used screws and plates and things. Why would they put screws in rather than just let them heal? Well, if you put a screw in or a plate in, then that means that the bones are going to be fixed together and this person is going to be mobile. And then the bones aren't going to separate again when they try and move about. And so that will increase the mobility, and it'll also promote healing. So patient will be more mobile, and this promotes healing. And you could possibly suggest a host of other things that will happen less, so less complications, e.g. if someone is immobile, then they'll get kind of bed sores. So bed sores, muscle wastage that we just talked about with the astronaut. So muscle wastage. And you can say blood clots, deep vein thrombosis, any number of possibilities that are sensible. That are sensible there. Part D. We have got some levers, and state the order. So we have got, if we've got here, we've got an effort, a fulcrum, and a load. So if we had, fulcrum, I know it's right near the edge. Let's put that there. We've got our effort on that side and our load on that side. Here we've got fulcrum, and then we've got our effort, and then we've got our load. So those are two different sorts of lever. The easiest one is our normal one, which is our first order lever, which is our elbow joint. And so that is when we are extending the arms. So that's our muscle, our triceps. So that one is the first order. And this is actually our third order, if you learn those. And if we had our second order, that's like the wheelbarrow, where these two are the other way around. So this is third order, and you've done a nice worksheet on levers. Explain why there's a difference in the type of lever represented when the arm is being bent and straightened. Well, let's talk about the biceps. So we're just going to explain where the fulcrum, the effort, and the load are, just like we've done here. So when the biceps flexes or contracts, let's put contracts or flexes, the effort is in the middle of the fulcrum and the load. So the effort is between the fulcrum or the pivot and the load. So the effort is between the fulcrum and the load. The effort is between the fulcrum and the load. And likewise in our triceps, contracts our fulcrum is between the load and the effort. So fulcrum's in the middle. So fulcrum in middle of effort and load. So you're just showing that you know what levers are in some examples. And then here's a little how science works, not a practical question. Suggest one feature of the human subjects that should be controlled. So just thinking that it's a bit like the astronaut one, so same age of person, same gender of person, same type of exercise, same length of arm, any number of things here. So typical things to go for is same age, gender, health, exercise, and so on. You can go on for quite a while there. And then the last little part is we've got our first order lever here, and you can see it's just a seesaw with force on each side of the seesaw, but the fulcrum isn't in the direct middle. And so using this equation you are asked to come up with the force needed over here. Physics and math students, you'll probably be able to just do this in your head, because what you're doing over here is this has got to, it says it's balanced, so hold the head erect, so that equals balance. So we just need to make these numbers add up on each side. So we've got the force times the distance. So the force times the distance on this side is 50 times by 2.5. And then when you do that, so 2 50s and half a 50, that's going to be 125. So you can do this in your head. And so that has got to equal 125 over here. The force is the question mark, and the distance is 5. So if you're doing this in your head, you can go, well, what times 5 is 125? So it's 25. So you could do 125 divided by 5. 125 divided by 5, 25. So 25 newtons. So 125, so let's do this properly. I'm using the proper 125 divided by 5 equals 125. So reasonably straightforward once you understand how the seesaw or first order lever happens.